California, where this all started, has a huge number of cannabis pharmacy and users. And the state government decided to try and do something about research to provide some real data on the benefits or otherwise of smoked cannabis, because as with MS or any other indication, there's very little scientific data out there. There's a lot of anecdote and a lot of belief, but very, very few clinical studies. So they set up this center and provided a research grant of about $9 million in the year 2000. And that's been dispensed to various investigators around the state. And there have been proper controlled clinical trials, relatively small scale, but properly controlled, of smoked cannabis. Now, it's quite difficult to do a trial of smoked cannabis because any cannabis smoker will tell you that the herbal material has a very characteristic smell, and it's hard to cover that up, and for the patient it's rather easy to tell the placebo perhaps from the active reefer. But nevertheless, it can be done. There are various ways of disguising that with flavoring agents. And the trials have been very useful. There are 5 studies completed so far. They confirm what we already thought, that smoked cannabis is of benefit for pain relief and spasticity. And some of the results in one of the California studies are shown here. This is a rather different design from the one I told you about before. This is a so-called crossover design. You take a group of patients who are first treated with a placebo, and then either treated again with placebo or treated with active drug. These are the placebo groups in each case, and this is the active drug. And you see this in this case, a significant reduction in the Ashworth scale, limb spasticity, the one that failed to show a result in the British MRC trial. But here's some real data. So that's been useful. And meanwhile, in this country, the situation still is that illegal herbal cannabis is used by many patients, particularly those suffering from MS, who absolutely convinced that it does good for their symptoms. We have the results of the clinical trial that I showed you earlier, which did show efficacy in an objective measure, and in all cases showed subjective benefit in terms of the patient's reports of improved pain and spasticity and sleep. And meanwhile, Sativex, the standardized medical product, has been approved as a prescription medicine by the Medicines Regulatory Agency just now in June 2010 for treatment of spasticity in MS, although it's not yet on the market. Still has to pass the hurdle of the NICE review and whether the NHS can afford it and does it really work properly. Nevertheless, it's approved, and that is really quite a significant milestone, because remember, this is still, at the moment, Schedule 1 material, intoxicant with no medical use. Now we're saying, even the hard-nosed regulatory agency is saying, it does have medical use. It's going to have to be rescheduled for medical use, and it will be on the market. And I suspect there won't be much abuse potential, because as a new prescription medicine, it's going to be quite expensive, and you can get much the same intoxicant effects by buying conventional street cannabis. So I don't think it's going to be a dangerous epidemic of Sativex abuse, but it is an interesting medical advance. And again, please remember, I'm not associated with this company. I'm just telling you the story. Meanwhile, towards the end of my lecture, the reason why we have cannabis receptors in our brain has puzzled a number of scientists. As soon as these receptors were discovered, they're clearly very elaborate pieces of protein machinery that respond to a plant product. Now, you had the same situation with morphine, another plant product, with opioid receptors, several different types of opioid receptors in the brain and elsewhere, recognizing an exotic plant chemical. Now, what's that all about in terms of evolution? We didn't evolve our brains surely to recognize plant chemicals. And sure enough, we didn't. We have our own naturally occurring opioids in the form of opioid neuropeptides, enkephalins, endorphins, etc. And as you see here, we have a whole family of lipids mediators which recognize the cannabis receptors in our brain and elsewhere. Some of these are both CB1 and CB2 active agonists. And there's not just one. The first one to be discovered was anandamide, which was again discovered by the Israeli group in large-scale extracts of animal brain. They isolated an active material that was active on the CB1 receptor binding site in brain homogenates. And Roger Pertwee in Aberdeen was the first pharmacologist to show that anandamide also had activity in a variety of the smooth muscle, in vitro smooth muscle preparations, such as mouse vas deferens, well known to classical pharmacologists. It was active biologically also in the same way as THC. So that was a real discovery. Anandamide was the first one, called anandamide after the word for bliss, or in Sanskrit, I think. Anyway, then lo and behold, we've got a whole slew of them. We've got 5 on this slide, and there are 8 different arachidonic acid derivatives, all of which have the same property of acting on CB1, CB2 sites. What a future generation of PhD students and postdoctoral graduates and scientists will show is why on earth do we have so many different ones? What do they all do? Why do we have different ones in different regions of the brain? All that is really a blank canvas at the moment to a large extent. But we can see pharmaceutically some interesting possibilities. If we can invent drugs to manipulate this endogenous, naturally occurring system, they might have some of the beneficial effects of THC or cannabis in a more focused way. For example, we know that these substances are largely degraded by either enzymic processes or tissue uptake processes. You can inhibit the transport process, or you can inhibit the enzyme. Pharmaceutical chemists are pretty good at making enzyme inhibitors, and this is already happening. And some of the inhibitors of the enzyme fatty acid amide hydrolase, FAAH, are already showing interesting pain-relieving effects in animal models. And you can see the parallel here with monoamine uptake inhibitors as antidepressants. By inhibiting the transport mechanism or the enzyme, you would prolong the actions of endocannabinoids only in those regions where the system was being active. These are not chemicals that are stored in nerve endings. They're more like prostaglandins. They're made on demand, and their synthesis is rapid. And since they're small, easily diffusible molecules, they reach their targets very, very quickly and easily. So those parts of the brain or the periphery where this is happening would be targeted selectively by drugs that were to inhibit their inactivation. Conversely, you could think about drugs that inhibit the biosynthetic enzymes. So there are a lot of possibilities there for the future, and many of these are being actively pursued already. And I'm sure we will see a new generation of cannabis-based, endocannabinoid-based medicines coming in the future. But Iverson's rule is that it usually takes 20 to 30 years between a laboratory discovery such as this and a new medicine being available for patients. So I don't know if I'll live that long to see this happening. But it's got tremendous potential, and that's just summarizing what I've already said. There's also the possibility of making metabolically stable analogs that would prolong the life of compounds like anandamide that are otherwise very rapidly inactivated. And there's a certain amount of interest in CB2. The CB2 receptor as an anti-inflammatory potential pain mechanism is being pursued also, and drug companies like the CB2 receptor because there's no danger of having a psychoactive side effect, because this is not associated with the intoxicant properties of cannabis. And finally, if you're very optimistic, you can think about all sorts of human conditions that might be treated by cannabis-based medicines. I'm not saying I believe in all this, but there are a number of indications I haven't had time to talk about. But anyway, I think I've finished, and I'll stop there. Thank you.